And Dr. Dayspring, we are live. Welcome.
All right. Hello, everybody. And we got a really cool topic tonight. And it's one that is just emerging because all of a sudden people are concerned about the encroaching epidemic of cognitive impairment and dementia as the population ages. And lipids, of course, play a gigantic part in that. So let's see. Here we go. So the brain contains the highest level of cholesterol in the body, approximately 20 to 25% of whole body cholesterol, and it synthesizes all of its cholesterol, which has a half-life of from 6 months to 5 years, compared to 2 to 5 days half-life of cholesterol circulating in our plasma. So once the brain makes a cholesterol molecule, you can see it hangs around forever. The brain keeps its cholesterol because it's the only way it can get cholesterol, and it is obviously such a cholesterol-rich organ. Overall, brain cholesterol turns over some 250 to 300 times slower than that in the circulation. The lipid lipoprotein systems of the brain are fairly independent of that in the peripheral cells, and the CNS cholesterol does not exchange whatsoever with other body pools of cholesterol. So we all measure cholesterol and other lipids in our plasma, and plasma cholesterol concentrations have zero relationship to what's going on in the brain. That is why there are people who go through long lives with very low cholesterol levels. We have pharmacological drugs that can blow plasma, LDL, and total cholesterol away, and that has nothing to do as far as injuring the brain in any way. So that is maybe the most crucial point you take home from today's lecture. Do not ever tell a person because whatever you're measuring in the plasma is low that it's affecting the brain. So the real reason I wanted to do this talk is because of this emerging epidemic of Alzheimer's disease. And if you don't know, it's highly related to the amount of cholesterol in the brain. So Alzheimer's disease is named after, guess who? Dr. Alois Alzheimer, a German psychiatrist and neurologist. In 1906, he observed and documented changes in brain tissue of a woman who had suffered from memory loss, disorientation, and other cognitive issues. After her death, he examined her brain and identified abnormal protein clumps, now called amyloid plaques, and tangled fibers, now called neurofibrillary tangles, which are the hallmarks of the disease. Amyloid is a general term for protein fragments that the body produces normally. Beta amyloid, and that's its abbreviation, A, the beta, is a protein fragment snipped from an amyloid precursor protein, the abbreviation of which is APP. And in a healthy brain, these protein fragments are broken down and eliminated. Yes, in a healthy brain. In Alzheimer's disease, there is an abnormal accumulation and aggravation of amyloid beta in the brain, leading to neurotoxicity, inflammation, synaptic dysfunction, and interaction with that tau protein. ApoE isoforms in ApoE lipoproteins can regulate the metabolism of amyloid beta by competing for the binding of LDL receptor-related proteins with amyloid beta in astrocytes. Two of several brain changes associated with Alzheimer's disease are accumulation of the protein fragment beta amyloid into the clumps that I just described, and the accumulation of the tau tangles. These changes are followed by damage and destruction of the neurons, termed neurodegeneration, which along with beta amyloid and tau accumulation is the hallmark of Alzheimer's disease. Plaques and smaller accumulations of beta amyloid damage neurons by interfering with neuron-to-neuron communication at synapses, and that's how our brain works. Inside neurons, tau tangles block the transportation of nutrients and other molecules essential for normal function and survival of the neurons. Although the complete sequence of events is unclear, beta amyloid may begin accumulating before abnormal tau, and increased beta amyloid accumulation is associated with increases in tau. So, one lipid we often don't talk much. We always talk about cholesterol, maybe triglycerides, but the brain is equally enriched with phospholipids. And just here's the history of phospholipids. You can see it was discovered in 1719 by Johann Thomas Hensling, analysis of brain, where he noticed, hey, there's ash in the brain, and the analysis of that ash revealed phosphorus. In 1793, a Frenchman, Antoine Fourcroy, described a fatty oil in brain tissue, which was later confirmed by another scientist. In 1811, Nicholas Valquelin performed the first complete analysis of the brain using state-of-the-art equipment, whatever that was in 1811, and he described the uniqueness of a fatty substance in the brain. In 1834, Jean-Pierre Courbe was the first to apply organic analysis to brain products, and he related the oil substance also to phosphorus, but also cholesterol, and he named the fraction brain wax. Finally, in 1847, Theodore Nicholas Gobley published a paper in 2 parts linking the fatty acid composition of egg yolk and viscous matter of the brain using a new data from carp eggs, and he called this lecithin. And that's what we call our major phospholipid even today. Gobley had many contributions to science, but he is totally remembered mostly for discovering the phospholipid lecithin. So let's get back to the brain and phospholipids. If you haven't heard of the term plasmalogens, it's like the predominant phospholipid that's up in the brain. And it's a class of ether phospholipids. For those who know biochemistry, R and R prime are 2 different groups, and they're attached by an oxygen molecule. That is an ether connection. And it plays a critical role in membrane function of the brain, heart, and immune cells. And cell membranes are what the brain is all about. No functional cell membranes, no good brain function. Growing evidence suggests that an ethanolamine plasmalogen is the subtype of phospholipids that has a very close association with Alzheimer's disease. Decreased level of these ether phospholipids have been commonly found in AD patients and were correlated with the cognition deficit. The potential mechanisms underlying the beneficial effects of the enolamine phospholipids on AD is related to the reduction of something called gamma secretase. And that's why I got into phospholipids, because gamma secretase is how you're going to tie in cholesterol to Alzheimer's disease. So again, for the biochemist, there's a plasmalogen up top compared to a normal phospholipid below that, and you see the double bond linking to the ether. All right. There's another very important category of phospholipid-like molecules in the brain called sphingolipids, and they contain a backbone of sphingosine bases, basically ceramide, a set of aliphatic amino acids that includes sphingosine. Ceramide is simply a sphingosine with a fatty acid. There you see it. Sphingomyelin is the attachment of that in a phospholipid-like molecule. And there are different types of sphingolipids, and you can see by their name, they're definitely going to be associated with neurologic tissues. So let me show you, pictures worth a zillion words. So you see the cholesterol molecules, and they locate between sphingolipids. And they really love each other, and they work together to create functional segments of the membrane called a lipid raft. And it's in those areas where you see that purplish, that's where the proteins are expressed on cell membranes of whatever tissue you're looking at. In the brain, it's going to be astrocytes and neurons. And it's the localization of proteins that determine what gets into a neuron or what gets expelled from the neuron. So it's critical to have the proper alignment of sphingolipids and cholesterol. And if you're going to have too much cholesterol, you're not going to have that correct spatial alignment. And if you have too little cholesterol, you're not going to have that cholesterol sphingolipid spatial arrangement. And you're going to have dysfunctional neuronic membranes. So cholesterol definitely prefers its buddy, sphingomyelin. So let me now tie in excess brain cholesterol to Alzheimer's disease. I think in the previous lecture, we let everybody know that cholesterol is absolutely essential for every cell in the body, so much so that every cell can make it. But excess cholesterol will crystallize and kill any cell in the body. And that's why you never want neurons to have too much cholesterol, or heart muscle cells, or any cell to have too much cholesterol, because those cells will die. So let's tie in excess cholesterol, which is found universally when you autopsy Alzheimer's brain, as to what's going on. Cholesterol levels in the brain are tightly regulated for physiological brain function, but mounting evidence indicates that excess cholesterol accumulates in AD, where it may drive the pathological changes. This seems especially relevant for the late onset Alzheimer's disease, as several of the major genetic risk factors associated with that are also associated with cholesterol metabolism. So here's the cartoon. Now you see this neuronal membrane up there, and the little black dots is the phosphorus head group, and the two legs that go down are the fatty acid tails of phospholipids. And you can see in that little orange segment, there is a specialized collection of single myelin and cholesterol. That's called the lipid raft. And you can see, because of that spatial alignment, all of a sudden proteins are being located there. And these proteins are, in pink, the amyloid precursor protein, and in the other rectangular or oval dark blue, gray, or light blue things, these are enzymes called secretases. So now you also see on this membrane up top, we're going to show you what happens when a membrane becomes cholesterol overloaded, or we're going to show you, it looks like, hey, cholesterol with a minus sign, it's cholesterol depleted. You never want a membrane to be cholesterol depleted. That would be a pathological membrane. That should say normal cholesterol, because that's what makes for a normal membrane. So the availability of astrocyte cholesterol regulates amyloid beta production by substrate presentation, which provides a potential explanation for the role of cholesterol-associated genes as risk factors for Alzheimer's disease. Astrocyte-derived cholesterol specifically traffics neuronal amyloid precursor protein, the APP with the light blue boxes there, in and out of clusters where it interacts with enzymes called the beta and gamma secretases, which you see on the right side expressed in the lipid raft. And you're going to see those two enzymes all of a sudden changes amyloid precursor protein into the little red box below it, amyloid beta, which you don't want to generate in excess. So the beta secretase enzyme initiates the amyloidogenic pathway by cleaving APP at a specific site, creating a fragment that is further processed by the gamma secretase, and it's this pathway that leads to the production of amyloid beta peptides, which aggravate and form the plaques with Alzheimer's disease. My friends, you are seeing how exactly too much cholesterol in a cell membrane, in a glial cell, or especially in a neuron, is going to generate what you don't want generated, amyloid beta. When neuronal membranes are loaded with cholesterol, amyloid precursor protein increasingly interacts with the beta and gamma secretases, generating more and more amyloid beta, ultimately resulting in plaque formation over time. Thank you, excess cholesterol in the brain. In low cholesterol membranes, I would rephrase that as in normal cholesterol membranes, amyloid precursor protein interacts with alpha secretase. And if you read that little yellow bullet point, alpha secretase cleaves APP in a way that prevents the formulation of amyloid beta peptides. This pathway is non-amyloidogenic and produces fragments that are thought to have neuroprotective properties. So never have excess cholesterol in your cell membrane in the brain. But can the brain synthesizes all its cholesterol. If the brain does have excess cholesterol, can it get rid of it? Stay tuned. The balance between these pathways is critical for brain health and dysregulation, such as increased activity in beta gamma secretases or decreased activity of alpha secretase, and it can lead to the accumulation of toxic amyloid beta peptides, contributing to Alzheimer's disease and other dementias. Nice diagram here. You see all of the cells floating around your brain matrix up there. And there are the cells in the brain are neurons, and every cell in the brain that is not a neuron is called the glial cell. But there's not many of them. If you see up top in the fine print, the glial cells are your astrocytes, microglial cells, oligodendrocytes, and something called ependymal cells. Now, what is between these cells that are all interacting with one another? That's not open space. That's brain interstitial fluid, which is a connective tissue called the matrosome. It's the brain interstitial tissue. And if lipoproteins are sharing anything with one another, including lipids, those molecules have to swim through the matrosome to get where they're going. Bullet point number 2, the brain does all of its lipid work without using blood vessels. No, it's not like astrocytes make a lipoprotein in the brain and they put it in your blood to go over to a neuron. No, the astrocytes create lipid-carrying particles and they swim over and then get internalized by the neurons. Here's the cartoon version. Look closely and you'll see the different glial cells there, as well as the neuron in the middle in tan. Notice the matrosome is all the space between these various cells. But notice up top, you see the ependymal cells. They are a very specialized cell with cilia on them that are the interface between the brain matrosome and the cerebral spinal fluid. But notice that green astrocyte has its legs wrapped around an artery. And separating the astrocytes from that artery is the blood-brain barrier. And that's not easy for things to pass through to get into the brain. In fact, most things don't. But there is also a brain CSF barrier. And that's pretty much how the brain expels stuff it doesn't want. Because once it's in the cerebral spinal fluid, it can go over to the choroid plexus, enter our bloodstream, and then be taken to the liver or wherever to be further catabolized and gotten rid of. So things, I go in the plasma, lipids go nowhere if they're not trafficked in the plasma. In the brain, lipids go nowhere if they're not trafficked in the matrosome. So glial cells are have other names, as you see there, are non-neuronal cells in the CNS, both brain and spinal cord, and they, unlike neurons, do not produce electrical impulses. They maintain homeostasis, form myelin in the peripheral nervous system, and provide support and protection for neurons. In the CNS, astrocytes, ependymal cells, microglia, and oligodendrocytes, their main functions are keeping the neurons alive. In the peripheral cells, the oligodendrocytes supply all the myelin that is needed to coat the cells. Ependymal cells, as I said, they are the interface between the cerebral spinal fluid and the matrosome. So the neurons, you all know this, they have axons and dendrites. They create the impulses that travel down them to wherever they're going. Neurons synthesize cholesterol starting in utero and in childhood. But around the age of 10 or 11, the neurons stop making cholesterol and they let the glial cells make it and send it over. Early in life, when we're forming, the brain needs so much cholesterol that every cell has the power to synthesize it. But you're going to see why neurons stop. So the astrocytes are by far the most numerous cell type in the brain, and their production does vary depending what part of the brain you're looking at. But it's the biggest part of the glial cells. And you can see they're star-shaped cells, and their main function is biochemical support of the neurons, but they also are the interface with the blood-brain barrier, deciding what can get in and some things what can get out. Astrocytes are the major source of cholesterol in the central nervous system in adults, as well as the major producer of apolipoprotein E, which is the lipid transporter protein in the brain. There is no ApoB in the brain. Microglia are a type of basically brain immunological cells. They're like the macrophages in the brain. So they support the immunologic function of the central nervous system. And they're incredibly sensitive to even small pathological changes. And their job is to rush to the scene of the crime and ameliorate that. So, including gobbling up tau and amyloid beta before it can become in a plaque and prevent fatal damage. So thank you, microglia. Here they are. Here's the #1 cholesterol-producing cell in the brain, the oligodendrocytes. And most of virtually all of the cholesterol they produce goes to form myelin sheaths. And we have a lot of myelin both in our central nervous system and the peripheral nervous system. And the other big part of myelin is phospholipids. So it's another happy mixture of cholesterol in betwixt phospholipid molecules. But there is that blood-brain barrier, and it represents a very important difference between the brain and other organs. No other organs have a membrane that is this strict, deciding what can get in and what gets out. Although endothelial cells in most capillaries are loosely aligned, the capillaries in the brain have tightly packed cells and form units along with a cell called periocytes, and they tightly wrap those astrocytes. Smooth muscle cells, astrocytes, microglia, and neurons. The endothelial cell of brain capillaries lack pores as a result of this complex tight junction system, and they have a continuous basement membrane which limits the transport of lipid molecules into the brain through the vesicular mechanisms, which is how lipids enter the cells in the periphery. So never confuse peripheral and central cholesterol metabolism. And here's another graphic. On the left, you see the blood-brain barrier. Starting as we go left to right, you see the arteries are wrapped by endothelial cells and those periocytes that are tightly wrapped that connect with the blue, which are the astrocytes there. And you see separating the astrocytes from the neurons as you go left to right is the matrosome. And then you see the ependymal cells that connect the matrosome with the cerebral spinal fluid. Once something's in the cerebral spinal fluid, it goes over through the choroid plexus and enters the bloodstream. So this is why it's easy to measure things in the cerebral spinal fluid, unlike sticking a needle into the brain and seeing what's going on there. So much of what we know about the brain is by analyzing lipids, lipoproteins, and whatever, tau, amyloid beta, in the cerebral spinal fluid. All right. So very interesting article, pretty long, talking more about the blood-brain barrier. So you see on the right are the little cartoons of the structures that you're going to see on the left. And you see there's a critical entrance protein, abbreviated as MFSD2A, and you see what its name is, which I won't repeat. And that things that just can't slip through the blood-brain barrier can be pulled into the brain. And you can see that, hey, there are, in their oblong, darkly colored purple or green apoproteins that can be pulled into the brain. And there are lipid molecules that either can come in or they bind to receptors that pull them in. But take a real close look and you see a little tiny HDL particle. It's that yellow circle with a couple of dots in it. And it looks like that HDLs can get through the blood-brain barrier and enter the brain. We're going to have a lot more on that later. But what is the only lipoprotein in the plasma that carries virtually no cholesterol? It's HDL particles. The average HDL particle carries 45 molecules of cholesterol. The average LDL, 2,500, and the average VLDL, 5,000–6,000 molecules of cholesterol. So even though a few of the smaller HDLs might get into the brain, they're not dragging cholesterol with it, and they are not a supply of cholesterol for the brain. Some lipids, such as free fatty acids, a component on triglycerides, but also in the phospholipid legs, can get through the blood-brain barrier and cross the blood-brain barrier. ApoE cannot cross the blood-brain barrier. It's just too big. These serum lipids can also act on receptors at the blood-brain barrier, which could include a transport for other things going on. So ApoE in the brain matrosome has some signaling effects as to what might get into the brain, but it's certainly not cholesterol. Serum lipids, including ApoE, cholesterol, and fatty acids in the brain matrosome can affect blood-brain barrier permeability. Serum lipids also have indirects, and this is where the triglycerides come in, because the fatty acids on triglycerides regulate or actually control leptin and insulin and ghrelin as to whether it can get in the brain or not. All right. You probably never heard of the G lymphatic. I know you all know what the lymphatic pathways are. So the brain lacks an identifiable, distinct lymphatic circulation, but it does clear extracellular proteins and extracellular solutes by cycling them to the cerebral spinal fluid through something called the G lymphatic pathway, and the G stands for glial. So a substantial portion of subarachnoid CSF cycles through along the wall of the artery. Up top in red is an artery, and it gets into the glial cell matrosome. It sort of goes to its base, and then it's picked up on the other side by the cerebrospinal fluid, and whatever is trapped in the G lymphatics then gets dumped into the cerebrospinal fluid, which gets dumped into the venous circulation. A better, closer look, artery on the left, and you see the connective bulk flow of basically water from the cerebrospinal fluid gets in, and it's sort of at the lowest level, the floor of the matrosome with the cells above it. It gathers the metabolites that they have released, and that floats in that cerebrospinal fluid, the G lymph, over to the, it gets expelled into the cerebrospinal fluid and makes its way into the venous system. So that is the G lymphatic system. Here's my cartoon of it. You see the plasma, and you see things can go back and forth between the G lymphatic system and through the blood-brain barrier, but also waste can be dumped into the cerebrospinal fluid for exit into the blood CSF barrier, through the blood CSF barrier into the plasma. The G lymphatic system is just a waste clearance system for the brain. Notice it works primarily during sleep, using cerebrospinal fluid to flush out the waste. And that's why the proper amount of sleep is so crucial to cardiovascular health and to brain health. So cholesterol synthesis in developing CNS is relatively high, but it declines to a very low level in the adult state, which is explained by a recycling of brain cholesterol. Hence, as I've already told you, cholesterol lasts forever in the brain. If the half-life is 5 years, by the time the last molecule goes, it's times 7, 35 years. In the CNS, unlike the periphery, virtually all of the cholesterol is unesterified, and you'll see where the cholesterol ester does exist transiently in the brain. In animals, most growth and differentiation of the CNS occurs in the first few weeks or years after birth, and then the cholesterol required for this growth comes totally from de novo synthesis. Currently, there's zero evidence that cholesterol can be transferred from the blood into the brain or the spinal cord. Again, stop worrying what LDL cholesterol tells you about the brain. In adults, the rate of synthesis exceeds the need for new cholesterol. Hence, the net movement is if there's too much cholesterol in the brain expels it. The processes that maintain cholesterol homeostasis are not static, but they change during early human development and later again in the aging brain. Cholesterol synthesis is highest in utero in the first decade of life, supporting all of the developing brain. And all brain cell types, including both neurons and glial cells, contribute to this cholesterol synthesis in utero and in development. And remember, after birth, most children have LDL cholesterols of 10 to 20 to 30 milligrams, yet they're making cholesterol excessively because they don't rely on plasma cholesterol. Once again, an adult brain cholesterol synthesis rates declines, and astrocytic cholesterol production ensures sufficient levels to keep the neurons happy. Third trimester of the pregnancy, the first 3 months, and then up until age 12 and then to adults. And you can see almost all the cholesterol is formed by the time you are in the low, I don't even want to call it the teenage years, just before the teenage years. And this is looking at top of whole brain weight, and then the bottom whole brain cholesterol content. And you can see it all occurs, almost all in the first 24 months of human existence. And then it keeps going until the adult, age 10, 12, when your skull is basically formed, certainly your brain size is formed. All nucleated cells in the body can synthesize cholesterol from acetyl-CoA. It's a very complex biochemical process, over 30 steps long. And once it gets to the first sterol, there are two pathways called the block and the Can-Dutch-Russell pathway that lead to the development or synthesis of cholesterol. And here you go. First, we go through a linear molecule, squalene 30 carbons, then cyclicizes and forms the first sterol precursor of cholesterol, linosterol. And then notice there are two pathways coming up here. On the left, the block pathway. On the right, the Can-Dutch-Russell pathway. Notice the block pathway goes through a penultimate, next to last sterol called desmosterol, and the Can-Dutch-Russell goes through lithosterol. And I show you the structures of those molecules on the right for the biochemists who are following my slides. The mevalonic acid pathway begins up top and it undergoes numerous steps. And once it's linosterol, there are two ways to get the cholesterol. And that's because the pathways are tissue specific. So to synthesize one molecule of cholesterol, do you realize it takes 36 molecules of ATP? Cholesterol biosynthesis is the most energy intensive anabolic process in the body. And that's why the brain hangs on to it and recycles it. The neuron doesn't want to waste 36 molecules of ATP, which it needs to set off action potentials in the synapses. So the neuron stops making cholesterol as soon as it can. It lets the astrocytes make it. They can use up their ATP, which has nothing to do with neuron ATP, and they can send their cholesterol over. So again, on the astrocytes on the left, the neurons on the right. And you can see there are two pathways of cholesterol synthesis. In the astrocytes, the predominant adult cholesterol-forming cell goes through desmosterol. Neurons early on, when they're making it, they go through the Cahn-Dutch-Russell pathway, which gets shut down around the age of 12, and neurons stop making lithosterol. They all rely on astrocytes to do desmosterol. So there's a lower rate of xenosterols and cholesterol synthesis in neurons compared to other glial cells. And you can see there can be some crossover, and you see it's the desmosterol pathway that rescues the Cahn-Dutch-Russell pathway. Astrocytes synthesize 2 to 3-fold more cholesterol than neurons. Oligodendrocytes even more, but they're selfish. They just keep it for the myelin. It is also believed in the mature state because of the great ATP expenditure, neurons synthesize very little amounts of cholesterol, and they rely almost totally on the astrocytes to provide it. So you see cholesterol, you see the penultimate precursors, lithosterol on top, desmosterol. You know what's good about them? We can easily measure them in the bloodstream. So if you measure lithosterol, which is the synthesis pathway used by the peripheral cells, you will know whether a patient is a hyper or a hyposynthesizer of cholesterol. But if you measure desmosterol, which is the brain pathway of cholesterol synthesis, the lithosterol pathway is not used in adults, you will have a biomarker that can give you a clue. Is the brain oversynthesizing or undersynthesizing or normally synthesizing cholesterol? And I'm about to show you that might be very useful information. And here we go. So this is, they took spinal fluid desmosterol and they measured it on the x-axis, and on the y-axis they measured plasma desmosterol. And what you're looking at here is the desmosterol cholesterol ratio, but the same graph occurs if we were just looking at the absolute concentrations. It's a straight line. That's a very good correlation. So desmosterol plasma level or the ratio in the same patients was found to significantly be decreased in those with Alzheimer's disease. Thus, this is the first study to say, doctors, if you measure desmosterol and it's low in a lipid drug naive patient, you have a great biomarker to warn you that Alzheimer's disease may be in the future. And what if you measured this with people who have E4 genotypes? Wow. Then you have a double whammy that this person is headed down the cognitive impairment route. And then hopefully, if you can find one, you can hook up with a preventive neurologist who knows how to advise these patients. And you should be doing this just like we want to measure cholesterol in young ages in the plasma. These type of biomarkers need to be measured at young ages in the plasma of those who carry E4 alleles or those who have family history of Alzheimer's disease. And here's the bad news. And you can see up top, we're going to look at the absolute concentration of desmosterol or the ratio in all patients, in men or in women. And you see the color differences there. And as desmosterol goes down, 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 the incidence of mild cognitive impairment is worse, as is the incidence of Alzheimer's disease. They go up. So you have an incredible biomarker to better help you evaluate such patients. Until now, we haven't had much. There are emerging neurologic biomarkers that you can also check, but this is a lipid biomarker. Wow. You do notice that it's more significant in women, but women get more Alzheimer's disease than do men. And this is looking at in Alzheimer's disease, the proportion of desmosterol was found to be less than that of virtually everything else in age-matched controls. The failure to detect sterol intermediates is an indication of no active cholesterol synthesis in the late stages of Alzheimer's disease. And you know why? Because there is so much cholesterol in the neuronal membranes that the brain shuts down cholesterol synthesis as a way to protect the brain. And hence, you see very low desmosterol in brain tissue, brain cerebrospinal fluid, and in the plasma. I could, you're lucky we don't have enough time. I could show you over 10 articles that stress the importance of desmosterol as a critical lipid brain biomarker. So let's get into cholesterol metabolism. There's cholesterol. Now, cholesterol can be converted to other molecules. And it's stored or transported in lipoproteins in an inert form where a fatty acid is attached, cholesterol ester. Of course, in steroidogenic tissue, cholesterol can be converted into steroids hormones. In many tissues, cholesterol can become an oxysterol. And do you see what differentiated? I put it in red. All of a sudden, there are more OH, hydroxy, oxygen molecules on the cholesterol. Now, they have many functions. In the liver, they're bile acid precursors. In macrophages and microglia, they are involved with immune function. But look in the brain, the predominant oxysterol formed in the brain is 24S. S, you see in the bottom center, that talks about the spatial alignment of the hydroxy group. Ignore that. But notice on 24S hydroxycholesterol, there's a hydroxy group at the 3rd carbon and a hydroxy group at the 24 carbon. Critically important, because you're going to see that's how the brain gets rid of any cholesterol that it doesn't want. How about brain triglycerides? They're present in the brain, but in infinitesimal amounts, because the brain uses glucose for energy, not fatty acids, unlike all the other cells in our body. The brain primarily relies on glucose. Under certain conditions, of course, ketone bodies for energy, not triglycerides or fatty acids. There's the triglyceride concentration in the brain, 0.65 milligrams per deciliter, which is 0.6% of the plasma concentration. So, and you know why there's no triglycerides in the brain? Because there's no way to traffic them. You need big lipoproteins like VLDLs and chylomicrons to carry chains. The brain, as you're about to see, has tiny lipoproteins. Triglycerides can be hydrolyzed at the blood-brain barrier, and the free fatty acids come right in and then be reesterified within the brain cells in astrocytes or microglia. Triglycerides do influence leptin and insulin signaling, modulate dopaminergic activity, and contribute to lipid droplet formation, occasionally in glial cells. So let's get into the brain lipoproteins. Discoidal spherical particles that have the diameter of 8 to 12 nanometers. What lipoprotein in the plasma has a diameter of 8 to 13 nanometers? They're called high-density lipoproteins. Now, ApoE is synthesized in the brain, the second largest producer after the liver. As I leaked before, the brain does not produce apolipoprotein B. So ApoE is the structural protein that wraps brain lipoproteins. And there you see it. And because of its small size, it would have a very low density because lipoproteins not carrying lipids sink in the centrifuge tube. Lipids make them float. And there you see an ApoE enwrapped particle that has the buoyancy density of a plasma HDL particle if you put it in the centrifuge. And the brain HDLs can have 2, 3, occasionally even 4 copies of ApoE. Like the HDLs in the plasma that have ApoA1, they can have 1, 2, 3, 4, 5 copies of ApoA1, which is a smaller peptide than ApoE. The main function of ApoE particles is to regulate lipid transport between neurons and the glia. And these particles facilitate what is called, guess what, HDL functionality in the brain. So here we go. There's an astrocyte. It's just made a bunch of cholesterol via the block pathway. Thank you, desmosterol. And upregulated is ABCA1 efflux transporters. It effluxes out the free cholesterol to a cholesterol acceptor protein, which is ApoE. It forms a discoidal type lipoprotein, which carries LCAT, which esterifies the cholesterol to cholesterol ester, a hydrophobic type of cholesterol, which goes to the center of the particle, and the particle matures into a lipidated ApoE containing HDL. But the astrocyte can actually construct the ApoE containing particle within its cytosol using acyl cholesterol acyltransferase. And then it just secretes the ApoE containing HDL particle. Show it again in slow motion. Here comes the ABCA1 cholesterol efflux transporter. Cholesterol is effluxed out and it's picked up by apoprotein E typically. I did stick an ApoA1 there, stay tuned. LCAT esterifies the cholesterol, forming a more mature, now circular, small HDL particle, which can even pick up more cholesterol via the ABCG1 or G4 transporter, and really makes a large brain HDL particle, which is exactly what happens in the plasma. And now let's look at the neuron. What is the neuron expressing on its properly constructed lipid rafts in the cell membrane? LDL receptors, LDL receptor related protein 1, scavenger receptor B1, and ABCA1. Every single one of them can be expressed at the liver in the periphery. But what are they doing in the brain? The LDLR and the LDL receptor protein, the ligand that attaches to them is ApoE. So they internalize the brain HDL particle. It goes into a lysosome and it gets destroyed. Cholesterol is released, picked up by the Niemann-Pick C1 and C2 proteins, and traffics through the cytosol. If you have a homozygous absence of NPC1 or NPCT2, that is a severe pediatric lipidosis. Now, there's the scavenger receptor, which can grab ApoE and internalize cholesterol ester, which gets deesterified to free cholesterol. If the brain wanted to get rid of free cholesterol, it could reesterify it, and you'll see what happens. So the brain, the neuron, has what it needs is a pool of free cholesterol, which is just trafficked over to its cell membranes where all of its work is done. But if the brain suddenly needed cholesterol, it could reactivate the Cann-Dutch-Russell synthesis pathway and would contribute to the pool of free cholesterol. It would only do that if for some reason the astrocyte was not sending off over enough cholesterol. Now, if a neuron has too much cholesterol, it could crystallize and kill the neuron, so the neuron can pump out free cholesterol using ABCA1 to ApoE, which can bring it back to the microglia or the astrocyte for good riddance. But the neuron is the only cell in the brain that contains the enzyme CYP46, 24S-hydroxylase, which creates that sterol I just taught you, 24S-hydroxycholesterol. But because it is a hydrophilic lipid, it can slip right through the neuronal cell, go down to the blood-brain barrier or the matrosome CSF barrier, and be sent right into the cerebral spinal fluid. Once 24S-hydroxycholesterol enters the plasma, it either rapidly binds to albumin or a lipoprotein, which returns it to the liver. It could also go right back to the astrocyte and be reused. So the cholesterol, via the enzyme cholesterol 24-hydroxylase, becomes the double hydroxy group hydrophilic lipid molecule 24S-hydroxycholesterol. And much of the cholesterol that's released from the brain in periods of too much cholesterol is in the plasma where it can be measured. And if you find 24S-hydroxycholesterol in the plasma, it is a great marker of brain origin, and it's a great marker of brain cholesterol toxicity. If the brain had a normal amount of cholesterol, it's not making or excreting 24S-hydroxycholesterol. So it's a useful biomarker as brain aging or a marker of Alzheimer's disease. So now you have another marker. We can measure the ApoE, we can measure desmosterol, or we can measure 24-hydroxycholesterol. Now you have three biomarkers of cognitive impairment or Alzheimer's disease. Bad news, 24-hydroxycholesterol is not available in the real world. It's a research test. but desmosterol certainly is, and ApoE genotyping certainly is. So what would happen if the brain all of a sudden was not making cholesterol? Well, there would certainly be no excess of cholesterol in the neurons, and so the neuron would not be making 24S-hydroxycholesterol. So this is why under periods of normal cholesterol synthesis, you will not find that biomarker. But what if there was suddenly some hyposynthesis of cholesterol? And this is why we know low desmosterol is a biomarker of Alzheimer's disease. It's being suppressed. So measuring low 24S-hydroxycholesterol or no hydroxycholesterol with a low desmosterol certainly tells you there is decreased synthesis of cholesterol in the brain. We're going to get to it because we're going to finish up with statins. But the single leading cause of decreased 24 desmosterol in the bloodstream and normalizing 24S-hydroxycholesterol are statin use. Now listen, there's too much cholesterol in people getting Alzheimer's disease, so you want to suppress brain cholesterol synthesis to a certain degree. But occasionally it can happen. You never want to over-suppress cholesterol synthesis in the brain. And now we have biomarkers that can tell us if you are using a statin and desmosterol is still at the lower end of normal, and if you could measure 24S-hydroxycholesterol and you couldn't find it, the statin is doing its job. Here's how 24S-hydroxycholesterol gets out. When it goes into the membrane, the fatty acid tails of the phospholipids want nothing to do with the hydrophilicity, and they separate. So synaptic vesicles, you can see on the bottom right, that's where the action potentials occur. In the adult state, neurons rely on delivery of cholesterol from the astrocytes. This outsourcing allows the neurons to save energy and use astrocyte-produced cholesterol for its lipid rafts. Cholesterol delivered to the neurons in this way supports synaptogenesis. Generation of 24S-hydroxycholesterol is an avenue that the neuron is saying, I got too much cholesterol, get out of here. Desmosterol would be suppressed. As you can see, the 24S-hydroxycholesterol goes back and it's sets off the nuclear transcription factors that regulate that. So astrocytes, but not neurons, express ApoE and secrete lipoproteins. On the other hand, neurons, but not astrocytes, express CYP46, which can help get rid of excess cholesterol. And it signals the astrocyte to stop making it. Now, let's wrap up these lipids in the brain with HDL complexities in the brain. So again, you see the cerebrospinal fluid, and you see the blood-cerebrospinal fluid barrier. You see the ependymal cells separating the matrosome from the cerebrospinal fluid. And you see the blood-brain barrier separating plasma from the matrosome. And the little gray, remember, on the floor of the matrosome is the G-lymph. So now let's talk about HDLs in the brain. So the first thing you see is, I've already taught you, the brain lipoproteins are ApoE-containing HDLs. So the one you see right in the center of your screen, that would be a normal brain HDL particle. It's enwrapped with ApoE. In the top right, you can see that functional HDL can interact with the G-lymph, with the microglia, and it can accept garbage from the neuron, including tau and amyloid beta. All right. Now, expressed at the barriers I just described is that scavenger receptor B1. And what is a ligand for scavenger receptor B1? I told you in the brain it's ApoE, but in the plasma, SRB1, HDLs carry ApoE in the plasma, but it's also a receptor for apoprotein A1. And this is how ApoA1, a very low molecular weight apoprotein, or even small HDL particles, can leave plasma and enter the matrosome. So now all of a sudden you have two types of HDL particles in the matrosome. ApoA1 containing HDLs that came from plasma, carrying very little cholesterol, so they're not supplying cholesterol, or ApoA1, which gets in. And ApoA1 can start accepting cholesterol from a cell, like a neuron or an astrocyte that wanted to get rid of it. But really, you know what it does? It jumps on the ApoE-containing HDLs. And it's basically, you now have complex HDLs in the CNS that contain both ApoE and both ApoA1, both of which can bind to those receptors I taught you on the neuron: LDL receptor related protein, LDL receptor, scavenger receptor B1, or the ABCA1. So bottom left, CNS HDLs contain mostly ApoB, but ApoA1 can be present. Now, if you have ApoE4 HDLs, you have dysfunctional HDLs. Top right, look at the neuron. It's trying to get rid of its amyloid beta and tau and even cholesterol to an HDL. And it could easily do that if that's an ApoE or an ApoA1, ApoE non-E4 HDL, or an ApoA1 HDL. If that's an ApoE4 HDL, dysfunctional, and you can't get rid of this tau and amyloid, and it just precipitates as plaque. So you can see HDLs, just like in the plasma, it's all related to their functionality. And a big part of the functionality of an HDL, as it is in the plasma, is its proteome. And in the brain, ApoA1 can rescue ApoE4 HDLs. So if we ever have a drug that can tremendously increase ApoA1, it may cross the blood-brain barrier, enter the matrosome, and rescue ApoE4 HDLs. Right now on the market, we do not have any drugs that raise ApoA1 significantly. Oh, a little bit, but you would have to raise ApoA1 into the stratosphere to send it into the peripheral or the central nervous system. So this is interesting. On the left in the small diagram, I show you a brain with a little circle that's been damaged. The neurons are damaged. Let's see what happens to ApoA1 in the central nervous system. So you can see going left to right, ApoA1 increases in the central nervous system, and that is associated with less neuronal damages. And you can see in the middle, there is far less beta amyloid deposition because the ApoA1 is gathering the amyloid and obviously getting it out via the G lymph or the microglia. And on the right, the disease score, their cognition is improving. Wow. So ApoA1 all of a sudden may be resurrected as could be good if we can tremendously increase it. But right now on the market, there is no drug that tremendously increases it. Statins, like 3-4%, nothing. Ezetimibe, very little. Bempedoic acid, very little. PCSK9 inhibitors, no. Maybe there's something coming that drastically raises ApoA1. So the studies in the past have shown ApoA1 can be a biomarker for various types of neuronal damage. And they've actually reconstituted HDL with ApoA1 in animals and seen neuroprotection. And would a mimetic peptide be delivered into the cerebral spinal fluid to increase ApoA1 HDL in the brain? Well, there's a drug coming that's going to do this, so fingers crossed. So what is the difference between the periphery and the brain? So we're going to go stepwise, top to bottom in the left column. With respect to biosynthesis, peripheral tissues, they make all the cholesterol they need. None of them require a delivery of cholesterol except steroidogenic tissue, and they get that delivery from HDLs. ApoB particles do not deliver cholesterol to steroidogenic tissues, which is why a low LDL cholesterol has no relationship to any steroid hormone. In the brain, almost exclusively the cholesterol is from synthesis, mainly from glial cells, mostly the astrocytes in adult, sometimes in neurons for sure, in utero and in kids, far less so in the adults. Lipoproteins. Wow, you got the whole family of ApoB and ApoA1 particles in the plasma. You know them. But in the brain, it's CNS lipoproteins with the density of an HDL. There are no particles that have the buoyancy density of an LDL or a VLDL in the brain. And there is no lipoprotein little a in the brain. Forward cholesterol transport is basically bringing the liver to the periphery, and that's HDL particles bringing cholesterol from the gut or the liver to steroidogenic tissues. Reverse cholesterol transport is both the HDLs and the ApoB particles, primarily LDLs, returning unused cholesterol to the liver or to the gut. In the brain, it's from the astrocytes and other glia as secondary contributors to the neurons by brain lipoproteins, which are ApoE containing particles or ApoA1 containing particles. Remember, the brain cannot synthesize ApoA1. Any ApoA1 in the brain has passed through the blood-brain barrier or the blood-CSF barrier. What are the apoproteins in the periphery? I couldn't even, it goes up to ApoM. I only listed a few there. But in the brain, not too many. ApoE, ApoA1. ApoJ is an inflammatory protein which helps get rid of garbage. ApoD is sort of a cytosolic transfer apoprotein. C1 and C3 are in the brain. Nobody really knows what they're doing there, but they're incredibly tiny, so they can easily slip through the blood-brain barrier. And of course, in the periphery, a little bit of C1, a little bit of C3 is perfect. Too much C3 retards lipolysis and causes big problems. We have yet to learn what's going on with them in the brain. What are the source of apoproteins? Well, it's easy. In the periphery, it's the liver or the intestine. And in the brain, it's astrocytes, followed by other glia, making much lesser amounts. Even a neuron, if it's stressed, can produce ApoE. And when does a neuron get stressed? When there's ApoE4. But the neuron has the same gene, ApoE gene, as the astrocyte, so the neuron would make ApoE4, which is basically committing suicide. Cholesterol turnover in the periphery, it can be stored as cholesterol ester. In the plasma, it's transported as cholesterol ester. Once it gets in a tissue, it can be deesterified. In the liver, it's created, of course, to bile acids, in the steroidogenic tissue to steroids, and in the skin to vitamin D3. The only organ that can eliminate excess cholesterol from the human body is the liver by secreting either excess cholesterol into the gut, which can poop it away, or by the liver converting it to a bile acid, which can be pooped away. How about the brain? Cellularly stored as cholesterol ester or converted to 24-hydroxycholesterol and excreted to the plasma via the blood-brain barrier. The glia, the neuron, can also efflux cholesterol out through the ABCA1 to ApoE and be returned to the astrocyte, which would suppress further cholesterol synthesis. Very interesting. My joke is, how does the brain get rid of excess cholesterol? Well, if you listen to me, you say, oh, that's easy, Tom. It makes 24-S-hydroxycholesterol, and it winds up in the liver. Okay. And then what does the liver do with it? It changes it into a bile acid, sends it to the gut, which you poop it away. So in essence, the brain gets rid of cholesterol just like every other cell in the body, sends it to the liver, gets excreted to the gut, and if it's not reabsorbed, and bile acids can be, it will be excreted fecally. So, there's the tie-in from the brain to the anus. All right, now let's finish up by talking about our drugs that are so widely used. And please go into today's Journal of the American College of Cardiology showing the incredible decrease in atherosclerotic heart disease that have been induced by statins. But that's a lecture for another day. We're talking about the brain now. So back in the year 2000, increasing evidence suggests cholesterol plays a role in the pathophysiology of Alzheimer's disease. That meaning hypercholesterolemia is a risk factor for AD. And for the longest time, it thought, oh, yeah, well, that's going to cause cerebral atherosclerosis and dementia. To determine whether patients taking statins have a lower prevalence of probable Alzheimer's disease, they designed a cross-sectional analysis comparing the prevalence of probable Alzheimer's disease in 3 groups of patients from hospital data. Entire populations of the folks in the hospital, the statin users, and patients using cardiovascular drugs that were not statins. And notice the statins that were examined in the year 2000 were the first 3 that came on the market. But the first 2 had by far the most use, lovastatin and pravastatin, Methacor and Pravachol. Simvastatin came later and was not used as much. It was too potent and people were afraid of it. So interesting what this study is going to show. 50,000 patients from the computer databases followed patients between the years 96 and 98. And here's what happened. And on the y-axis is the prevalence of Alzheimer's disease per 1,000 cases. And notice the prevalence of diagnosed AD in patients using lovastatin and pravastatin was 2/3 lower than the entire patient population of those older than 60, with a significant p-value. Now scratch your head. There was no reduction in the people using simvastatin. Now, that's impossible. So that means there were so few people using simvastatin, they just couldn't generate what was believable statistics. Notice all the other cardiovascular meds used at that time, ACE inhibitors, diuretics, beta blockers, etc., had no impact on the incidence of Alzheimer's disease. And there were a few maniacs who were actually using lovastatin plus pravastatin. I mean, that is unconscionable nowadays, but back then people were putzing around. But it worked as far as reducing Alzheimer's disease. Remember what type of study this is. This is an observational post looking at hospital data, not the strongest evidence. But they did conclude there is a lower prevalence of diagnosed AD in patients taking two different statins. And while one cannot infer causality with this, the study certainly reveals an interesting association. So about a year or two, when statins first came out, nobody was worried about the brain. Everybody was just worried about, God, let's reduce heart attacks and strokes and bypasses, etc. And they were doing that beautifully. And then they started, let's look at other things that maybe lowering cholesterol works. And here's a hypothesis that the statins would do very good in the brain. So in 2003, we continue in an article published in the Archives of Neurology. 3 statins lowered levels of 24S-hydroxycholesterol by about 20% without affecting the levels of apolipoprotein E. This may be beneficial in the treatment of Alzheimer's disease through a direct effect on brain cholesterol. Remember, Alzheimer's is too much cholesterol. So certain statins, by lowering cholesterol synthesis in the brain, which would diminish 24S-hydroxycholesterol excretion, that would suggest the statins are doing what they should be doing, avoiding cholesterol toxicity in the brain. Statins may influence brain cholesterol metabolism directly because they are able to penetrate the blood-brain barrier, regardless of the hydrophilicity or lipophilicity. Look, if you're doing a pharmacokinetic study and you give 1 dose, lipophilic statins get in easier than hydrophilic. But once you're on a statin and you're in steady state, every statin on the market invades the brain. So if you're prescribing statins, whatever your favorite statin is, it is certainly getting in the brain and inhibiting cholesterol synthesis to a variable degree. Neurons, like all other cells in the body, must have a continuous supply of unesterified cholesterol, and this sterol must come from either de novo synthesis within the nerve cells or uptake from the extracellular environment, meaning CNS lipoproteins. Alzheimer's disease is a neurodegenerative disease caused by amyloid beta, and amyloid beta correlates with cholesterol and the deposition of plaque. So now we're up to 2021, and there's a lot of statin data that can be analyzed post hoc. So here we go, published in the Journal of Clinical Lipidology. What is the association between statin use and cognitive function? A systematic review of randomized clinical trials and observational studies. And cut right to the conclusion. The review confirms and extends prior systemic reviews in that they do not show a negative effect of statins in the brain. The consistent data from all these randomized control styles that examine statins and cognitive function should be reassuring for clinicians who prescribe statins for those that increase CVD risk. More importantly, these data provide reassurance for your more elderly patients that you are not screwing up cognitive function with continued statin use. Okay, and I think that's probably the opinion that is still held by most. But again, this is post hoc analysis of randomized trials or analysis of observational data. Along came Lou Culler, an epidemiologist at the University of Pittsburgh, again published in a Journal of Clinical Lipidology, and he wrote an editorial, Statins, Lipids, and Dementia. He says, look, meta-analysis is only as good as the individual study. The trials of statin therapy to reduce dementia or AD do not support a prominent role for statins in reducing AD or all dementia, nor that statins increase the risk of dementia. The quality of the studies were just not that good. They're not prospectively designed randomized control trials, which would be needed to ultimately answer this question. But I think we can all take home, in the overwhelming vast majority of people, that you're writing statins to reduce heart attacks, bypasses, and strokes. You are not causing dementia. And remember, statins are significantly lowering LDL cholesterol in the plasma, but that would have no influence whatsoever on the brain. It comes down to statins inhibiting cholesterol synthesis in the brain. Are they doing it? Not excess. Stay tuned. So in 2023, lipid lowering risk and the risk of dementia by the European Society of Cardiology. And they look, as you go from looking at all the studies, yes, a green check, you're reducing atherosclerotic cardiovascular disease, but dementia, they put a question mark. And in gray were the positive trials, and the pinkish were the not so positive trials. So they said, look, based on compelling mechanistic evidence suggesting a detrimental role of neuronal cholesterol in the development of Alzheimer's disease, it certainly has been hypothesized that statins are neuroprotective. Therapy has been increasingly linked, however, to short-term neurocognitive effects, such as amnesia episodes and memory, by case reports and surveys that even showed up in a few of the clinical trials. Of note, statin-associated amnesia episodes early on and memory deficits were typically reversible once you stop the statin, which would suggest a causal role between drug exposure and cognitive detriment. Anybody who's prescribed a statin know an extreme minority of patients come back with brain fog. And if we stop the statin, it disappears. To me, that suggests the plausibility that in some people, statins do oversuppress the cholesterol synthesis in the brain, and probably a statin is not the wisest drug to use in such a case. That's all a hypothetical assumption at this point. In 2012, the U.S. Food and Drug Administration issued a black box warning outlining the potential link between statin use and cognitive impairment, which was based on people writing letters to the FDA. That black box has since been removed. Conclusion. Contemporary evidence tends to support that lipid-lowering therapy with statins does not significantly alter cognitive function in the short-term trials that have been done. Long-term studies greater than 10 years of follow-up are pending, so stay tuned. And finally, in 2023, they had to put this to rest, and this is the American Heart Association statement. The risk of hemorrhagic stroke associated with statin therapy in patients without a history of CVD is non-significant, and achieving very low levels of low-density lipoprotein cholesterol in the plasma has nothing to do with hemorrhagic stroke in the brain. So I wish people on the internet would stop spewing that nonsense. Relevant data for the primary statin and non-statin lipid-lowering trials were reviewed, including basic trial demographic data, baseline lipids, on-treatment lipids, looking at all the references. In the summary, from randomized controlled trials, epidemiologic studies, and genomic studies, supports the use of statins and non-statin therapies to improve cardiovascular outcomes by aggressively lowering LDL cholesterol, parenthesis ApoB, and there is no lower threshold that is dangerous to the brain. So if you want to use combo therapy with a PCSK9 and make an LDL-C10, the Ebbinghaus study showed you can't hurt the brain by doing that. There, for the naysayers who want to read all these studies, you can pull down this article, and there are all the studies they reviewed. My goodness. So just published this year, Statin Use and Dementia Risks, a systemic review and updated meta-analysis. The findings now underscore the neuroprotective potential of statins in dementia prevention. Despite the inherent limitations of observational studies, the extremely large data set and the detailed subgroup analyses enhance the reliability of our results. Future randomized clinical trials, of course, are necessary to confirm these findings and enlighten clinical guidelines. Just published today. How appropriate for tonight's lecture in the Journal of the American College of Cardiology. The impact of cognitive impairment on cardiovascular disease. Dyslipidemia, and this is a long article, it gets into a lot of things, but I'm reading you the lipids section now. Concerns regarding the potential negative effect of statins on cognitive function have been alleviated by a systematic review of the literature, which showed no significant association between statin use and adverse cognitive effects. And they give chapter and verse in this paper of all the trials if you would like to go and read it. Some data even suggest potential cognitive benefits as is seen in a meta-analysis of greater than 7 million patients. The STARI trial and the preventable trial are now undergoing looking at statins in elderly people to clarify once and for all the use of statins for primary prevention regarding cognitive health. There is no debate about primary prevention on atherosclerotic heart disease with statins in men or women. Statin therapy generally appears safe in older adults, and more data on the primary prevention in very older cognitively impaired individuals are forthcoming. So stay tuned. So now, statins and brain fog. And this is the answer you're going to get if you go to AI and ask, what is brain fog? Brain fog is a potential side effect of statin, though it's relatively uncommon and still somewhat controversial. The FDA has acknowledged reports of memory loss and confusion associated with statin use, labeling them as non-serious and reversible. As I told you, the black box warning is bye-bye. These symptoms, often described as brain fog, can include forgetfulness, difficulty concentrating, and mental sluggishness. Anybody who has written a significant amount of statins to save lives knows that a few people are going to come back and say, Doc, whatever this statin is doing, I'm having these neurologic symptoms. So to me, I raise a hypothesis. Could that be maybe the patients where statins are significantly depressing cholesterol synthesis and immediately causing these neurologic symptoms? Of course, if you stop the statin, these symptoms go away, which suggests the brain started synthesizing normal amounts of cholesterol again. So there probably are some people who, even though you might want to use a statin to lower ApoB and LDL cholesterol, that you can't use them because you don't want to suppress cholesterol synthesis in the brain. And so what? Then you would either use a lower dose of the statin or forget the statin and go with ezetimibe, bempedoic acid, PCSK9 inhibitor, and other drugs that are likely soon coming. So remember this slide where, cognitive impairment and Alzheimer's disease are associated with low desmosterol, the ratio or the absolute concentration. So here's my hypothesis. By far the most common cause, if you're measuring serum desmosterone, comes back, they're likely to be on a statin. But this is why you should check desmosterone before you start a statin. Because if it's low before you start a statin, you know the brain by itself is suppressing cholesterol synthesis, and that is a big biomarker for Alzheimer's disease. If you haven't already done it, immediately do an E4 genotype on that patient. And if E4 is there, that's a big warning sign that maybe, since I do have to lower ApoB big time in AD patients, because it contributes to AD by cerebrovascular disease and other mechanisms, maybe I need to use lower dose statins or other lipid ApoB modulating drugs, of which we have right now. So hypothesis, can desmosterol concentrations be used to diagnose abnormally low brain cholesterol synthesis or to monitor for statin-induced oversuppression of cholesterol? And it's up to you. The labs that do sterile measurements are the Boston Heart Lab, and LabCorp sends out the specimen to the Mayo Clinic, which analyzes sterols. And the test in LabCorp is called plasma sterol levels. In Boston Heart Lab, it's called the cholesterol balance test. So there's much more to tell on that story. I had great fun doing the last 3 lectures. I thank my buddy Ron Grisanti for inviting me here to speak to the FMU. I can be contacted on Twitter. Please join the over 40,000 followers, and you will continue lipid lipoprotein education. Medical professionals may want to connect with me on LinkedIn. So I love this Peter Max diagram, which is happy lipiding because we're helping the heart. But I should ask Peter Max to do another drawing and sneak the brain into this graphic. So good night, everybody. Thanks for listening.
Thanks, Dr. Dayspring. I appreciate it so much. I want to thank everyone for participating, and I wish everyone, wherever you are in the world, to have a good evening or a good day. Dr. Dayspring, we're both on the East Coast. I want to say good night, and I look forward to connecting real soon. Take care of yourself.